Hey everybody, welcome back to another DNA Master tutorial. Today I'm going to be showing you guys how to blast all of your genes, how to put them in HHPred, and how to blast on Phagesdb as well. So blasting your genes is probably the most important step or the most strongest evidence in determining what your gene actually does, determining the function of your gene. So these things are very important, and I'll show you guys how to do them. So in last episode I showed you guys how to do the start site, the start codon stop site, stop codon gene orientation, the SD score, the SCS section, as well as the gap and the longest open reading frame. So today I'll show you guys how to fill out the blast section, the HHPred section, the blast P section, and the additional notes with the Phagesdb website. So what exactly is BLAST? So according to Wikipedia here, basic local alignment search tool. So what this is doing when I say BLAST, it actually is taking the amino acid sequence of your gene, which is shown here, and it's pretty much going through the entire gene, one amino acid at a time, and comparing it to a database of information that has already been input by other people and other scientists along the way, and it's coming up with results that match or are very similar to the sequence that you have. So it's very important that you not only know how to input your data into the BLAST tools, but it's also important that you know how to interpret them. So I'm going to teach you guys that today. So the first thing you have to do, this isn't in any particular order, you can do these in any order you wish, but I always do it in this order because I personally think it's easiest. But the first thing we're going to do is go up here to the BLAST tab. As you'll see, if you guys remember, back when you started your annotation, you actually ran BLAST for all of your genes in your genome. And what that did was send out the amino acid sequence of every gene in your genome. It sent it out to the BLASTP server, and it actually brought back all of those results in a nice little chart here, table. And it actually brought them back into the program DNA Master itself. So the first thing we're going to do is go read those results and place it back in the notes section here. So once again, click the BLAST tab. All you have to do is click over here where it says export. We're going to hit export. That's going to make the program generate this nice little note document here that comes up with all the hits from the BLAST results. So this is hit number 1, and this is the function of the first hit. So it's a lyse-M domain protein found in Arthrobac phage princess trina. All we're doing now is we're looking to see if this is a good result, and there's 3 things to keep in mind when looking for a good result. The first thing I'm going to talk to you guys about is the E value. That's what you want to look for, one of the things you want to look for. E value, the closer you get to 0, the better. And in this case, it's actually 0. So 0.0 times 10 to the 0 is 0. So you can't get a better E value than this, which is really extremely strong evidence that this is pretty much amino acid by amino acid, the same exact sequence as the thing you input. So this is an extremely good result. Another thing you want to look at is the query match here, and you want to see how it relates to the target match. So you can also see them here. So this is what we pasted in the top row. This is our amino acid sequence, the top here, and then the bottom row here is the target or the result. So this is princess trina phage sequence, and this is our sequence. And as you guys can see, amino acid by amino acid, they are the same exact sequence from the start to the finish, as you guys can see here. So you're going to look here at the query match and the target match, and if they match 1 to 1, that's even a bigger piece of information. That's even more evidence that these things are very good, very well aligned, and that you can actually use this result in the annotation of your gene. So the last thing you want to look for is the species that it comes from. So obviously we're annotating a bacteriophage, so you might not expect to find, a Homo sapien gene within a bacteriophage. So if this said like Homo sapien protein or something like that, you would obviously it's not the gene that you're looking for. It's very unlikely that the sequence from a human gene matches exactly to a phage gene on such a large scale. This is 222 amino acids. So you want to look at the species, the E value, and the match. If all those 3 things are looking good, we're in good shape. If this was like a 3 to 1 match, but it matched fully throughout the rest of the gene, you might have to go back and reconsider your start site, because sometimes you're off by just 1 or 2 amino acids, and that's matching these alignments here 1 to 1 is strongly suggested. So if it doesn't match 1 to 1, it's it's suggested that you go back and try to reevaluate your start site and see if there's a potentially better start site. So in this case, all three of these things look good, and we're going to go ahead and use this result as our as evidence. So what we're going to do is copy here from the right of the colon here. We're going to copy where it starts the lysem domain protein, which is the function, and we're going to end it here after %align. We're going to do Control C, which copies, and we're going to close that out. We're going to head back to the description, and we're simply going to paste it into our blast section here, and that's it. That's all you have to do for the blast result. So as you can see, it gives the quality control process, or the people working at the quality control, it lets them know all of the information without them having to switch over and export the table themselves. So that's all you have to do for that one. That's a pretty easy one. The next thing we're going to do is blast on the BlastP server itself on the website. We're going to blast on Phagesdb, and we're going to also use HHPred. So to get your amino acid sequence, we're going to go to the protein tab up here. We're going to do Control A and then Control C to copy, or you can just select all, right click, copy. And we're going to go to the Phagesdb website, the HHPred website, and the BlastP NCBI website. So literally all you have to do to get to these websites is you can Google NCBI blast, and it'll be the first result here. That will get you to the blast. You're going to click the protein blast because that's what we're doing, protein, and then we're going to open up HHPred. Again, we'll Google, all you have to do is do HHPred, one word, and it'll be the top result. And then for Phagesdb, it's obviously just phagesdb.org. And then we're going to go to the blast tab here and then hit BlastP, and it'll bring you here. So let's go back to the NCBI. First thing we're going to do, guys, is we're going to paste our amino acid sequence in there, and then that's all we have to do. We're just going to hit blast. Now these things take a while because going through a large database, as you can imagine, takes some time, especially if your protein is long. You know, if it's a couple hundred base pairs or longer, it's going to take a little bit of time. But this will give you the time since submitting, and it will refresh every couple of seconds, as you can see down here. So give it some time. Likewise with HHPred, we're going to paste it in here, but you have to remember an important step for HHPred. So this here is the database section, and what we're actually going to do is select another additional database. So we're going to not only blast it on this PDB70 database, but we're going to scroll down a little bit, and we're going to select PFAM-A as well. So you have to hold in your control key or the command key if you're using a Mac, I believe. So hold the control key down and select PFAM-A as well as PDB70. As you can see, they're both selected here. So we're going to go ahead once we do that and submit the job, and likewise this will take some time. Fortunately for us on Phagesdb, it's a very quick process. Literally all you do is paste it in, hit blast, and look at that, we have results already. So I'm going to go ahead and explain these results, and by the time I'm done explaining them, these will be finished as well. So that one already actually finished, but let's start with Phagesdb. So first I'm going to explain to you guys the color coding that's going on here. As you'll see in this little graph thing, all of these lines represent the length of the alignment. So the longer the line, the better aligned it is to your amino acid sequence. So we want to see full length lines, and as you can see, we see a lot of long lines here, which is good. And towards the bottom, obviously, we have some poor results, and they're shorter lines, and they're different in color. So as you can see here on this scale, the red is a very good alignment score. It means 200 or more of the amino acids aligned with the thing that you input or the result. So and we'll see here less than 40 matched with these results down here. So we want to see red, and we want to see long lines, and that's what we'll see up here. So what we're going to do, if you mouse over it, you can actually see the results pop up here. So it says mouse over to see the results. So we're going to do that, and you can see the first one is Humpty Dumpty draft, and we'll keep going down. We'll see some more. So what we're going to do is just scroll down a little bit for now. We're going to see a little table of information here. So again, we have the e-value column. This is, like I said, the closer to 0, the better. This is e to the minus 129. So if you can imagine 10 to the minus 129, that is ridiculously close to 0. It's practically 0. So these results are very, very good. Past that, I mean, e to the minus 63 is still extremely good. until we get down to about 2 times 10 to the minus 4. That's a pretty poor result. We generally don't consider results that are above, that are higher than 1 times 10 to the minus 5. So this is a bigger number than that. So we generally don't consider that. And likewise, as you'll see here, they just continue to get bigger. Eventually down here we have 8.2, which is a horrible e-value, and we usually never consider those results. So if you look over here, these are the name of the species that that matched your result. This is the function, and this is how many aligned with, how many of their amino acids aligned with yours. So if you remember, our gene was 222 amino acids long. So all of these results have practically 100% alignment. You know, as you can see here, 222, 221, 217. They're pretty close. So the other thing to look at is the name here. So if it has draft in the name, that means it hasn't gone through the quality control process yet. It still hasn't been annotated, which may explain why the function of the gene is unknown. So generally don't look too far into things that have draft because they're not annotated. They probably aren't going to be helpful because they will have an unknown function. So we're going to scroll down this list until we see a result that does not have draft in its name. So top hat draft, that's gone. Scavito draft, that's gone. Rosy Posy draft, that's gone. Princess Trina 23. So Princess Trina does not have a draft in its name. So that's the first phage that's made it through the quality control process. And as you can see here, it has a function and its match is 100%. So this is the result we're going to look at. If you guys remember, that was actually the phage that we copied into the BLAST section from our BLAST results as well. So you could put the same phage result in twice. I generally try to avoid it because, you know, if you listed this in the BLAST results section, you're practically going to list it, and then you list it again in the Phagesdb section. It's kind of redundant, you know. It's you already listed this as evidence, you already listed this phage gene as evidence, so it's not really worth putting in. However, like I said, you can choose to use it if it is some more information that you think would be interesting for the quality control people. But I'm going to keep scrolling down the list to find another one that doesn't have draft. So Cabriz, Humpty Dumpty, Edgar Poe, Comboy, which is actually the phage I'm working on right now, Chubster, Chocolate, and ARV1. So ARV1 doesn't have a draft, so that's the next annotated one that has a function, and it can be used as evidence. So I'm going to actually use this result here as evidence because although it doesn't have the best score, as you know, it doesn't have 457, it still has a really good e-value. It still matches 100%, and it matches the function of the Princess Trina that we did earlier. So that's going to give us some more evidence for the QC process. So all I'm going to do is scroll over here. I'm going to click this little hyperlink to the score, and it's going to take me down to the result. All you have to do now is copy everything from right of this little arrow here. So we're going to copy right, and we're going to scroll down. We're going to go to the end of positives, and we're going to right click, copy, or just Control-C copy, and we're going to insert that into at the bottom here where it says additional notes aligns 1-to-1 with previously QC annotated homolog phage ARV19 peptidoglycan binding protein lyse M. We can get rid of the 222 because it says length up here. We'll just consolidate this a little bit. So again, this is really good evidence because not only does it show up here that, hey, it matches with a phage called Princess Trina 1-to-1, e-value 0. This is a really good result. It's a lyse M domain. But it's also found in another phage, ARV1 gene 19, and it also is a lyse M peptidoglycan binding protein. So this is going to be extremely good evidence as well as this, and that's the ultimate goal. If you didn't list Princess Trina, we're going to note to the QC people below, we're going to say maybe put a period here, and we're just going to say that it also matched with the Princess Trina phage as well. So we're going to say also matched 1-to-1 with previous QC annotated phage Princess Trina 23, but I did not list that here because it was listed in the blast section. So that will let them know that Phage Princess Trina 23 was the best result, but you went with this one because it provided more evidence and because Princess Trina is already listed up here. So that's pretty much it for the Phagesdb section. So now we're back onto the blast section, and as we'll see here, this top section only comes up in certain situations, and when it does, you always have to note it. So whenever there's a putative conserved domain, you always want to note that into the notes of your blast piece section. So what this is pretty much a region or a group of amino acid, a sequence of amino acids that is conserved amongst many genomes, and whenever it's found, it's always the same function. So, and they're listed right here. So we'll see superfamilies, multi-domains. We're just going to click the top one here. So lyse-M superfamily. And when you click it, it opens up a new tab, and it's going to explain what this lyse-M superfamily is. So in the top here, in this first result, we're going to see a lysine motif is a small domain involved in binding peptidoglycan. So that's pretty much what we've been finding, but this is even stronger evidence. So it's only a subsection here of the gene, but it's definitely domain, it's definitely present because it's a conserved domain, and we definitely have to note it. So we're going to just copy it through here. All right, we'll go back to the note section, and in the blast P, we'll say we found the name of the superfamily or the conserved domain, which in this case was the lyse-M superfamily. We're going to put a comma, then we're going to paste what we just copied just to give a little bit of a description to them. And we're also going to go back and we're going to note the interval here, 170 to 220. We'll go back and paste it in. And then we're going to note the E value. So we're going to do E value colon 5.03 times 10 to the minus 3 times 10 to the minus 10. So 5.03 E minus 10. And that's all we have to do for the blast piece section. So that's definitely good that we found a conserved domain, and we noted that here, and the QC process will see that. Now if there was no conserved domain that we found, this box up here wouldn't show, and you'll just see results. And when that happens, all you do is kind of click the top result. It'll take you down here, and you're going to go through the first couple of results to see if they not only agree in function, but if they are good results, again comparing the E value, the alignment, and the species. And as you'll see here, these are all pretty darn good results. We'll see the VB underscore ARTM ARV1 phage, we'll see Princess Trina again, and then we'll go down, we'll see Brent, which is also a pretty good result, and Barrett Lemon, lyse-M. We're ultimately seeing a lot of lyse-Ms, lyse-M, peptidoglycan binding protein. So peptidoglycan is the molecule found in bacterial cell walls, so it would make sense that a phage has a protein that will bind to that molecule or polymer and will allow the phage to maybe connect onto the bacteria and inject its DNA, something like that. So there's definitely a lyse-M. It may even help in breaking down peptidoglycan so that the phage can insert its DNA. But ultimately, we know this gene is involved in breaking or degrading the cell wall of the bacteria. So say if there were no putative conserved domains or superfamilies found, and we just had these results, all we would do is go down, scroll down here to see some of the top results, and we're going to again compare the E value, which is the expect value here. We're going to compare the alignment 1 to 1, as we'll see that's what we want, and the species here. So we'll see this is from a phage, from Arthropactor, which is good. So again, we'll just go through and we'll see kind of the general consensus of the top results, and we'll see throughout the board, 1 to 1, great E values from Arthropactor phages, and they're all coming to a consensus that this is a lyse-M, or it has a lyse-M domain or peptidoglycan binding protein, and ultimately that is good, that all of the top results are deciding on the same function of a gene. So we pretty much know the function of the gene at this point. So now at this point, if we see that this top result, if the top blast P result is actually the same result as what we found in DNA Master, and as you'll see that is the case for this gene, so we'll see the Arthrobaccterphage Princess Trina result, the lysem domain that we pasted in earlier. It's actually the same thing as this. All we have to do is go back into DNA Master and we'll just say something like top result is the same as the blast result from DNA Master. That's all we would have to say. Like I said, if there was no superfamily or conserved domain and the top results matched. You could also find, as I mentioned earlier, more evidence by scrolling down. We'll see we didn't use this phage yet, so we could copy this result and paste it in to the section here. All we would do is copy up to the gaps and just maybe, up here, we'd copy that, paste that in, and then we'd copy this, paste that in, and say that it matched 1-to-1. And that's the BLAST-P section. So we're going to end it with the HHPred section, so we'll go to HHPred. And likewise, you'll see similar layout as the previous website. So we're going to see the length of the alignment and the colors. So this is the top result. We're going to click that and it's going to take us here, phage P2 GPU. This family consists of several bacterial and phage proteins of around 130 residues in length. Again, we're going to look at the top results, not just the first result. We're going to look at through the other ones. So we'll see the result 2 has a lysem domain. Again, this is what we've been getting. So we'll see the E value here, 4.2, E minus 8, and that's definitely something we can use as well. And let's see here, unknown function, can't really do anything with that. Auto-lysin, which is kind of related to the lysem domain, but generally we're just going to stick to the top couple results. So HHPred can be a little confusing sometimes because the databases are varying, they're unique, and they're different from BLAST-P. So this is where you have to decide. This information here is kind of vague. It doesn't tell you anything. It says it could be involved in tail assembly, but it's not really coming to a consensus. It's like I said, it's just kind of vague. Although it is the top result, I'm not going to go with this one in this situation. I'm going to go with even more evidence that it's a lysem domain, and we're going to use the number 2 result. So again, good E value. It's not a good alignment, but in this case, it's nothing really to worry about. The information here says it's about 40 residues long, and it is found in a variety of enzymes in bacterial cell wall degradation, which is pretty much what we came to, the conclusion we came to earlier. So I'm going to use this result. Now on HHPred, there's also another category that we look at, and that is the probability. So we have to report the probability as well. So the probability, we don't accept anything generally below 90%, and you can see here this is a very good probability, 98.72%. Like I said, if it drops below 90, we generally don't consider those results, but it does happen sometimes. We're going to copy maybe the first sentence here, or a sentence or two involved in cell wall degradation. We're going to copy that, and we're going to find the HHPred section, which is here. Going to paste that in there. We're going to consolidate it, and we're going to go back and copy this code here, or this sentence here. So starting from probability, we're going to copy it all the way over across to some probs, and we're going to just paste it into the line there. That's all we need for HHPred, and that's pretty much it for this episode. So if there were no good results, say if there was a horrible E value, horrible alignment, and it was a gene that is not related to phages, we would just say no significant results, as you'll see in this Famirator category, but we haven't gone over that yet. So we would just say no significant results if we blasted things and we got horrible results. Just because it's the top result doesn't mean it's good. It could have an E value of 10. So if they're all bad results, just write no significant hits. But that's pretty much it, guys. Thanks for watching. As always, if you have any questions, let me know. I'll be definitely willing to help you guys out. But I hope you guys learned something, and thanks for watching.